Uh, Good morning, everyone. I My name is Vikas Arora. I represent Infocart India, and I would like to welcome you all. Thank you all for giving us this opportunity. So we are having this online meeting, for onboarding and orientation of various digital solutions subscribed by pharmacy colleges in the state of Andhra Pradesh. So I would like to thank for trusting us, for giving us this opportunity, believing in us and our services. And I promise you, on behalf of Team Infocard, on behalf of the three products which we have given access to, you have subscribed it through Jawaharlal Nehru Technical University, Anantapur, and we will be providing you best of our services. So today I'll be just quickly sharing my screen so that we can be on the. Can, so we will be talking about the products, where what all we have given. So currently there are three products, and different institutions have opted for different products. So first product is Lexi Drug. Lexi Drug is a premium drug information service wherein access is given through web application into the college and through mobile application for the students who are going to the hospital. Now, with the help of this mobile app access, they will be able to make use of it onto the patient bedside when they are being given, they are being posted into the hospital, when they are being. So we will be having a demonstration of Lexi Drug. So I would like to put it like this. I will not be wrong when I'm saying that we are the only company, only organization who is dedicated for providing various products under one umbrella. So we are providing you drug information in the form of Lexi Drug. We are also offering my PharmaRx, which is a documentation software. So as of now, the institutions have not opted for it, but it is also available. If any of you are interested, especially the institutions who are looking for NAC inspection, there they ask about digitization of the processes, many of the institutions. So it is important to share over here that the product has been developed along with JSS Mysore, the reputed JSS College of Pharmacy. And it is being used by various reputed institutions like Deccan College of Pharmacy in Hyderabad, KL in Karnataka. So these are the institutions who are using our this service of documentation software. Next software which we are providing to the consortium is MyCalPharm. MyCalPharm is a simulated animal experiment software which is being used. We are currently providing you 40 plus animal experiments across the campus, so you can make use of it. And lastly, we are providing access to check for flag. Now, when we are talking about the master courses, or when we are talking about the PhD courses, in fact, in case of PharmD also, where dissertation needs to be done, now the authorities, whether it is AICTE or it is the university, they take keen interest into the integrity of the documents. What content is, if it is the original work by the researcher, or it is being plagiarized using the easy availability of content over the internet. Now, to have a curb on it, to make a again to get quality documentation or original content from the institution, we are providing this, the check for flag. I proudly say it is Make in India initiative, and our core agenda, our core thought is to provide you quality information resources at affordable price. Lastly, Before I hand it over to my colleague for starting the demonstration, I would like to talk about uh, Journals Card. Now, Journal Card is an initiative wherein we will be talking about hosting of Indian journals, and here we will be providing you the access journal to the Indian journals into the field of pharmacy. Here is the agenda on your screens. So I'll be now handing it over to my colleague, Ms. Ruchi. She will be talking about up-to-date Lexi drug, which is the drug information service. It would be followed by Mike Alpha. It would be followed by Mike Alpha, which is the simulated animal experiments, would be taken by Dr. C.R. Patil. Dr. C.R. Patil is the author who is the for this Mike Alpha, and he has agreed. So I requested him that many of the colleges would be joining. So he agreed. So he will be. So it is always a pleasure to have a demonstration from the author itself. So he will be demonstrating, and it would be followed by a demonstration of check for plague from by my colleague Ms. Surin. She will be talking about the check for plague, how we can make utilization. So this is I'm just starting the session now, and I would thank you once again to all of you for joining in. So Ruchi, you can start the demonstration of Lexi drug. So over to you, Ruchi.
Good morning, everyone. Thank you, sir, for giving me this opportunity to represent Lexi drug. Lexi drug is a point of care tool which helps in taking right clinical decision for the right clinical decision on the patient bedside. So I'm just sharing my screen. Kindly confirm that my screen will be visible or not. So my screen is visible to everyone?
Yes, Ruchi, it is visible.
Yes.
Okay, fine. Thank you for the confirmation. So this is a up-to-date Lexi drug, which is having the two type of access. One is web access, which I am showing on my screen, and second one is a mobile app, which is used by the student at the on the patient bedside. When they are in the ward, hospital, they can just take out their mobile and have the information on the patient bedside. So talking about up-to-date Lexi drug, we have different kind of modules over here. You can see Trezor IV compatibility, interaction, drug ID, patient education, calculator, and more clinical tools. Here is the search bar. Apart from this, help and training resources are there. Training videos, quick reference guides go there. I'll just start with the drug name. Let's suppose I'm searching for a drug, ciprofloxacin. So here you can see the result is there. Ciprofloxacin systematic is there. So here is the result for ciprofloxacin systematic, where you can see Lexi drug multi-international, all the drugs, pediatric, neonatal, everything is there. So I'll simply click on ciprofloxacin systematic over here. And the result is here itself. Starting with monograph, images, adult patient education. Let me take with the brand names. If I want to go for a brand names, I can just search the country name. Let's suppose I'm searching for India, but all brands are there for ciprofloxacin systematic itself. So I'm clicking on India and the brands are here itself. Next is brand name international. International non-proprietary name is there. Apart from this, pharmacology category is there. Next, we can come up with the dosing, which is very important point over here. Dosing for all the population, for adult, older adult, altered kidney function for adult. Just showing you, this is a oral immediate release, administrator as soon as possible, less than 6 hours until next schedule itself. So this is oral extended release. Dosage adjustment for concomitant therapy itself. Dosing for older, you can refer to the adult dosing itself. and dosing for altered kidney function for adult itself. Dosage for altered kidney function adult itself, you can see over there, oral immediate release, oral extended release, and in through IV also you can give the dosing itself. Next, we have altered liver function for adult itself. The hepatic dosing recommendation are based on the best available evidence and clinical expertise. Senior editorial team itself. Initial or dose titration in patients with pre-existing liver disease or dosage adjustment in patients with chronic or worsening hepatic function during treatment itself. So here are the dosage for pediatric also. Whatever information you are getting in up-to-date like drug, that is all reference based. Here you can see the clinical consideration is also given. Fluoroquinolones should not typically be used as first-line therapy due to their adverse effect profile and risk of development of bacterial resistance. Only used in situation when there is no safe and effective substitute available. Next, we have altered kidney function for pediatric also given for infant, children, and adolescent. Through IV or oral immediate release, there is no dosage adjustment provided in the manufacturer's label. However, the following guidelines have been used by some clinicians. Next, we have an altered liver function for pediatric itself. There are no dose adjustment provided in the manufacturer's label used with caution in severe impairment itself. Next, we have a calculations. Next, we have a usage label indication is also given for children and adolescents. Treatment of complicated urinary tract infection and probably due to, although effective ciprofloxacin is not the drug of first choice in children itself. So in infant, children, adolescent, and adults itself. For adults, treatment of the following infection when caused by substitute bacteria. So all the information label indication is given over there. Off label for adult also given. Level of evidence information is also given. Classes related monograph is given. Administration through IV injectable table. How should we do the administration for oral also given over there. Next, we have a storage and stability. How to store this drug. Preparation for administration for adult is given. Preparation for administration for pediatric is given. Next, we have a IV compatibility, which is really a different module we can go itself. Next, we have a warning and precautions. What all contradiction is there? Warning and precaution is also given over here. For special population is also given. Older and adult consideration is there. Pregnancy, reproduction, pregnancy, and lactation consideration is there. Next, we have a breastfeeding consideration. Big drugs in pregnancy and lactation is also given. And what all adverse reaction is there. Next, we have a patient and therapy management. What all monitoring parameters are there? Nursing practice point, which is a new point just come over here. What all preparations is there? Dosage forms consideration for US, Canada. Pharmacology and pharmacokinetics mechanism of action is also given over there. Now we can come up with the interaction part. So I simply click on interactions. In interaction, we can take more than 3 to 4 drugs to check the interaction. Let me take a drug name amiloride. Next, I can take aspirin, which is a common drug. Let me take hydrochloride. Let me take Remexor. Apart from drug, I can take herbal products also like green tea. Not only drug, we can take food product also. Let me take grapefruit. I can take even ginger or garlic. If I want to add, that can also be added, just showing you itself. Next, I will take a drug named Botulism. So this is all drug which I have entered, and let's suppose a patient is having an allergy of alcohol itself. And simply click on analyze button and the result is here. Here you can see 4 type of options are here. A means no known interaction, B means no action required, C means monitor therapy, and D means consider therapy modification, and avoid X means avoid combination. So let me go with the interaction. Here is the result. Hydrochlorine and remdesivir, they both are avoid combination. So why it is avoid combination? I can simply click over here. So here is the summary and the patient manager. Hydrochlorine may decrease the therapeutic effect of remdesivir, severity, major reliability. Patient management is there. Concomitant use of hydrochlorine and remdesivir is not recommended. So discussion is there. In the footnote, it is the reference is given there. If reference will be open, hyperlink will be there and you can go to the article also. But this is paid article itself. Next interaction, I can go over there. Autism and green tea depend on dose. So here it is written, the interaction is likely of greater concern with consumption of large quantity of green tea, more than 7 cups per day itself. So consider therapy modification. Green tea may decrease the antiplasic effect of autism. Patient management is given, discussion is given, and in the bottom you can see the footnotes are there. This is all reference over there. This is a PubMed article. I'll just simply click over here and I can go to the article itself. So this is the article. Next, we can go for a next interaction. Let me take amiloride and autism. So here is the summary. Patient management. Blood pressure lowering agent may increase the hypertensive effect of hypertension associated agent itself. Moderate reliability rating intermediate low itself. Patient management is given. Discussion is given. And in the bottom footnote, you will get the references there. So this is interaction. Let me take you another module of Tresol IV compatibility. Here, let me add the drug name heparin sodium. And let me take one drug name levofloxacin. And let me take this drug to normal saline. And I can simply click on analyze button. So here are the results: incompatible, uncertain, compatible. So these all three options are there. Here I can see it's written incompatible. So why it is incompatible? So I can simply click over here and the results are there. There are 2 studies given over there. I can click on one study. And the best part is there, whatever reference you are getting, you can take the print or you can save in the computer also. So this is a study period, container, physical compatibility, storage condition, methods, chemical stability, and citation also. Like this, I can go to next study also. We'll check another compatibility. Let's suppose levofloxacin is a compatible. So I can simply click over here and go to the studies itself. So here are the study, physical container, physical compatibility, storage condition, method, chemical stability. And in the citation, there is a reference given over here. I can simply click over here and see the reference-based article itself. So this is IV compatibility. Next module, we have a drug guide. Let's suppose a patient come and he can say that I don't know about the drug, but I have some idea about the drug itself. Let me take an imprint. It is having an imprint of 500 mg and dose in tablet form. Let's suppose their shape is round, color is white. That's all the patient knows. Nothing about manufacturing label and the product name. I simply click over here. So here are the generic name and the brand name of the drugs. With this result, you can find out the drug name itself. If you want to know by generic name or brand name, both you can check it itself. Next, we have a patient education. In the patient education, I can browse by condition, procedure, discharge instruction, healthy living, natural products, adult medication, and pediatric medicine. Let's suppose I have to search about any disease. Let's suppose I will take about hypertension. So you can see the result. The result are there: high blood pressure in pregnancy, high blood pressure emergency in children, medicines, what all medicines required. So all the information for discharge instruction, healthy living, and condition also. I simply click on this leaflet, high blood pressure and pregnancy itself. So information is here. You can take the print or you can save the in the computer also. More than 9,000 leaflets are there. Apart from this, if you want to go for alphabetically any condition, let's suppose, browse by category itself. Let's suppose we have to take a procedure alphabetically. How to use ear drop. So here is the result. You can see the basic information is given there. Apart from this, pictures are also given where you can take the print and give it to the patient also, which helps the patient to do the procedure itself. Next, we have a calculator. In the calculator, BSA, BMI, all the information is there. In the more clinical tools, we have a toxicology. Let me take a poison name. Let's suppose I enter morphine. So here is the result. So you can get the monograph, toxicology alerts, background, clinical presentation. Mechanism of toxicity is also there. Their diagnosis, exposure control, what all treatment require, stabilization, decompensation, patient disposition. So all the information is there. Next, we have a drug comparison. Let's suppose I have to compare more than 2 drugs. What all monograph is there, their dosage, their administration. Let me take drug name. You can take more than 2 also. I'm just taking a 2 drug comparison. So clicking over here, I can get the drug comparison by monograph also. Their box warning, their administration, their dosing, each and every information you can compare over there. Like this, you can compare data view also. Next, we have a new drug review. So let's suppose from 2014 till the date, you will get all the drugs which is approved by FDA itself. Let me take for August itself, new drug review. So here is the new drug. Their monograph is there with their brand name, pharmacology category, medication safety issue, usage, mechanism of action. Pharmacokinetics is adult data unless noted itself. Their pregnancy consideration, breastfeeding consideration, drug interaction is also given. So all the information related to this new drug, even their images also given over there with their brand name, label, imprints, dosage form, strength. Even you can go for their patient education leaflet also. So this is all information about lazy drug. Now I would like to invite Vikas sir. If you have any question answers, Vikas sir will be clear that.
Thank you for giving this presentation, taking a live tour of the Lexi drug. So definitely, if anybody has any questions pertaining to this particular product, service, Lexi drug, you can ask me. Before that, I would like to add a couple of lines from my side onto this particular service. So it is a global standard, means it is being used in US or Middle East or Africa or European countries. 100 plus countries we have usage of this particular, and the beauty of this is that it is being updated on a daily basis. So we have a big editorial team, 200 plus people who keep working on getting the curated information from various resources and provide you onto the cloud basis. Now, how it is going to be helpful is that now we are providing access over a mobile app and into a single mobile app. For drug information, you can use it, interaction, you can use it, IV compatibility, you can use it, calculators, you can use it. And the beauty of this app is it works offline too. Sometimes we face challenge. Now, just now I got a message from Dr. Padmanabha Reddy. He said that there is some challenge with the internet due to rain, and we all face that. We all face that at some point of time. So, Lexi Drug, the mobile app, has been designed in a way that the data gets downloaded into your device, and it will give the results even when internet connectivity is not there. Yes, you have to update it as in when you get into a internet connectivity, whensoever your Wi-Fi gets enabled or you get your 3G 4G connection back. So this is we have developed. Now you have a tool, the students have a tool, the faculty has a tool. When they are being posted into the hospital, they can make their role more meaningful because now they have Lexidrug, a reputed name. If they can have a better connect with the doctor, because we understand the challenge is there. Doctors may not value us. But now when we provide them quick information, reference-based information, information which makes their life easier, avoiding any kind of interaction or ensuring better patient safety. So that is where we come into picture. So yes, that was all from my side. So Ruchi has already explained it. So we have provided you access. It has been activated on web. We have also given you a authorization code along with the manual with which. So if you face any problem, you can reach out to us. And meanwhile, you can start using it immediately. Our core idea is to ensure optimization. Now the institution has decided to take this service. So the value should be given to the students, to the faculty. That is what our idea is. Yes, coming back to any queries pertaining to Lexi drug, so I'll be happy to answer. Anybody can unmute and ask any question they are having.
Hello, good morning, sir.
Yeah, good morning, sir.
This is Jairam from Vijaya Institute of Pharmaceutical Sciences for Women, Vijayawada.
Right, sir.
Sir, what static IPs of this for this software?
So, sir, there are, okay. Let me explain it. This is pertaining to the access of Lexidrug. Okay, so Lexidrug, we are providing access into two ways. One is the web access, wherein you can provide your static IP. We can get it whitelisted, validated into Lexidrug software and through their server. In any time, you will be reaching out through that particular IP. But you have to ensure, sir, that the IP should be static. The IP should be fixed. It should be public. It shouldn't be the dynamic which keeps changing. Otherwise, you will face challenges every time the portal will not allow you to enter. So if you have a static IP, you can check with your IT team or you can get us connected with them. We will be happy to make them understand what is required. And then you need not to use username password. Anytime you will be coming, any computer which is coming through a public IP will be given access because that IP will be whitelisted, validated into Lexidrug server, sir. So that is one part. Simultaneously, we are also providing mobile app access that has nothing to do with the IP. It would be governed by the authorization code that has already been shared. That is allowed for all the students and faculty, sir. They all can get the app download and get it validated to start using it. So I hope, sir, I have answered your query.
Yes, sir. But whatever the static IPs I was given to this Lexidrug, now we are going to change.
No problem, sir. Even if you are changing it, you can let us know. You can let us know. We will get the new IP activated, sir, and we will get the old one removed. So that all, sir, this is like you are changing something. We will be means that is not that you have taken one. So for the rest of the year, you will be using that IP. We understand that sometimes we change the service provider due to that the IPs get changed. So that is okay, sir. So once we will conclude, sir, I will be giving you contact details of the dedicated account manager for each product. And you can reach out to us at any point of time, sir. We will be happy to assist you.
Okay, sir.
Okay, sir. Thank you, sir.
Hi, Vikas. This is Satish from Vidya. Can you hear me?
Yes, Satish sir. How are you?
Yeah, very good. How are you doing?
I'm good. Nice to hear
nice
you
to
at
see
least.
you. Yeah. Now, I have a question for you. See, if people
Yes.
are logging based on the web access,
Yeah.
at any point of time, how many people can log in at the same time, such that it doesn't say that the limit is reached, or I mean, like
So,
at any point
right.
of time, how many people can access on the web platform with
I
one
understand.
username password?
I understand. So, when we have this, you call it as one is the IP access. Okay. When we validate the IP address, then honestly speaking, we do not have any limitation. 10, 20, 30 is fine with us. So nobody has complained to us that, okay, you have validated our IP, you have whitelisted our IP, and we are facing any challenge. So we haven't faced that in last 12 years. I'm working on this. Now coming to the other part of it, which is the username password based. So as of now, the arrangement has been done. Since we are providing mobile app also, so we have made an arrangement that the username password can be used at least in 15 to 20 computers at a time, at any given point of time, simultaneously. The keyword is simultaneously. Satish sir, I hope that answers your query.
That answers it, because my question is, because my students work in multiple locations in different hospitals and at the college at the same time. So
Right.
because they are logging in different IP addresses, I cannot log an IP address. But my question is, if more than 50 people have already logged into the account, is there a way I can, suppose it is not allowing more people to log in, is there a way I can remove the old logged in credentials and give access to the new people who log in, or will it, after
No,
the
sir.
limit is
It
reached,
won't.
what will happen? That's my question.
It won't work like that, sir, because the control is not with us for this logging out everyone and giving a new setup. But what I understand and what I humbly suggest as a solution to your query is, for students' point of view, sir, because they are posted into hospitals and they go to multiple hospitals. That also I understand based on what course they are doing and whatsoever the reasons are. So when we are giving you dedicated mobile app access for each and every student, then I don't see any challenge, sir. Why we? Because even in you, even if you are using it in a computer, the facility of taking it onto a patient bedside is not available, sir. So I humbly request that better we should like push it for a mobile app access to the student, and then sir, even if you will make each and every student a PharmD course available, means let's say you have a 6-year course and you have 30 students ka batch in each 30 students in each batch, then you can make it available to 180 students of the college. That is what we have made an arrangement for. Satish sir, does that
Yeah,
make sense to
that
you?
answers it. Yeah. My
Right.
only
So
thing is, what will happen if suppose 50 students have already logged in? Now somebody wants to log in. It
so
won't
sir,
allow them.
we want so if, let's say sir, we have made an arrangement of 200 200 let's say logins through mobile app, sir. Let me make it is through mobile app. So mobile app, you can have everyone. We are not restricting it to 50. Let it be 180 plus another 10, 20 faculty for your college. We have allocated 200 mobile app access to your institution, sir. Means to every institution, sir, who is the part of this consortium. Does that make sense, Satish sir?
Yeah, it makes sense. So what you mean to say is the mobile access doesn't count towards the other access. So this web access is different, mobile is
Actually,
kept
sir.
separate.
Actually, sir. So,
Okay.
sir, we take it as two ways. One is the practical utility, which is mobile app, sir. And one is the compliance point of view. If somebody is coming, no, though many colleges they show that the mobile access is there, but sometimes if the compliance point of view, an inspector is coming and he's asking whether you're having access to the drug information service or not, then you can have that. Or you are doing sometimes we are not accessing mobile and we are doing some work onto the computer. In fact, at times I also use WhatsApp onto my web application. So we have made provision for both, but from a student or mobile app access point of view, we have even given provision, made provision for the first year student. Means from year 1 to year 6, 30 into 6, 180 students, another 10, 20 for the faculty point of view. So you have access for, or you have a provision for accessing 200 mobile apps simultaneously for each college. And thanks you raised it, because now it is clarified to all the participants who are attending this that what mobile app access is all about. So I really appreciate that you raised this point. Thank you, Satish sir.
Thank you, Vikas.
Anyone else? Any more questions? Yeah.
My name is Sheikh Saddam Hussain from Sugarpeta, sir. Gokul Krishna College of Pharmacy.
Sheikh
As you
sir,
said,
I
it is
can.
available as mobile app. Does it support
Yeah.
offline?
Yeah, please carry on.
Sir, you said it is available as mobile app. Does it support offline?
Yes. Actually, your voice is not very clear, but what I could understand from that you're asking about the app availability. So yes, it is available on Android as well as iOS. If that is the question, and if I have answered it correctly, I'm really sorry, your voice was not very clearly audible.
Support offline. Does it support offline?
Again, I'm really sorry. Sir, can you please use the chat box?
Asking about like whether the app is available offline, sir. Offline database.
Yes. It is available offline. Thank you, Srinivasan sir, for making it clear. So yes, the mobile app is available offline too. So the data gets downloaded. In case of a mobile app, the data gets downloaded into your device, handheld device, and you can access it. You can make use of it. It will give the result even when the internet access is not available. But it won't work every time. Why? Because once the data, you need to update it. So for a limited period of time, let's say in some time it will work. But as soon as any update is there, then you need to update the content. So that is how it works. Dr. Sheikh, I hope that answers your query.
Yes, sir. Yes.
Thank you. Thank you for this. So if there are no more queries, one more thing, sir. Since the colleges have opted for these resources, so we will make sure to conduct demonstrations, means training programs. Now, demonstration is done. So training programs on a quarterly basis. So expect us to do a training program kind of thing with what new has been introduced, what more features are there on somewhere post Diwali, around Diwali, we will conduct one more. Means we will keep conducting these sessions so that we can also learn from you, and we can also make you understand what more we have added to the product, how you can do the optimization. And if there are no more questions, sir, I would like to invite now Dr. C.R. Patil, who is the professor of pharmacology in R.C. Patel Institution of Pharmaceutical Sciences. A very good friend of mine, like a elder brother, he's to me, and he will be demonstrating he's the author for one of the authors for this Michael farm and the animal simulated experiment. I really thank him for accepting this invite and coming in, joining in for taking us to the tour of Michael farm portal. So over to you, Dr. Patil.
Yeah, thanks a lot, Vikas sir. Am I audible?
Yes, sir, you are. You are, sir.
Thanks for this opportunity. I'm thankful to Vikas sir and his team in Pocat India Private Limited for this opportunity of demonstrating Michael Farm to Andhra Pradesh teachers, pharmacy teachers. Good afternoon, Reddy sir, Padmanabh Reddy sir. Thanks for joining these particular demonstrations. So let me tell you that it is an endeavor, means whatever I'm going to demonstrate you now is an endeavor of two professors, one from medical field and one from pharmacy field. So from medical field, there is a veteran pharmacologist Dr. R. Ravindran, who was working on simulation development since 1980s. We can say that he is a pioneer in field of development of pharmacological simulations. And then, like his distant disciple, I saw his softwares, and then I thought that for pharmacy students, there should be something that we can create, or a pharmacy teacher should create. And then I tried to develop something for pharmacy students. So whatever I developed, I got some idea from Dr. Ravindran sir's initial efforts to teach experimental pharmacology to his students. Now, this software is quite important these days because at undergraduate level we have been totally prohibited to use experimental animals, and even though it is an unfortunate thing, but still, if we see the kind of experiments which we are performing, there was a scope that those details of pharmacology could have been taught using such simulations. So let me share my screen. Hope this screen is visible to all. Vikasa, can you please confirm, is it visible? Okay.
Yes, sir.
Yeah, thank you. So this MyCalPhan is a sincere effort by two teachers to create simulated experiments in pharmacology, where whatever we have gathered across the years, we wanted to demonstrate to our students, and we wanted to help them understand experimental pharmacology during the era of ban on use of experimental animals, and this is going to remain there. Okay, so this is our portal through which, this is the website through which we are providing MyCalPharm. There are reasons why people should start using this, because this is a wholesome software where you will be getting video demonstrations, you will be getting simulations, students will be interacting with the screen or the animals which are present on the screen in the simulation. Teachers will be able to set examinations, teachers will be able to demonstrate and help the students to really understand what they're learning in the theory classes, and then how experimental pharmacology is helping, it is reinforcing the learned material through proper simulations. So there are many experiments. For example, let's take this effect of drug on heart rate and blood pressure of a dog. This is a classical experiment. It's when we are teaching autonomic nervous system to explain how different drugs, how different neurotransmitters are having distinct effects on blood pressure or heart rate. You cannot do it by conventional method using chalk and board kind of teaching. Okay, there you need a simulation. Again, if suppose you want to tell your students, say UG or PG students, how you are dissecting out anacoccious muscle from a rat, which is a very minute tissue, there also you cannot do it even in the wet lab practicals, because to a batch of 20 students you cannot demonstrate that minute kind of isolations or tissue isolations. Again, many times it is not possible that we will be having all the good qualities set up in our academic institutions. So even there, this software is going to help us. Now let me take you through few of the experiments. This is a very classic kind of experiment where Dr. Ravindran sir has tried to imbibe all the intricacies related to effect of drugs on muscles of iris as well as on intraocular pressure. So this demonstration is also important, where teachers will be very efficiently telling their students how various drugs can affect sensibilities, means if we are considering about local anesthetics, how we can evaluate local anesthetics using rabbit eye experiment, how we can check effect of adrenergic drugs on intraocular pressure, and all these things have been very nicely designed here. They have been very nicely simulated. So when we are studying effect of drugs on rabbit eye, there are different kinds of reflexes that we have to check. The first reflex is response to light reflex. Okay, so see, what I did is I just picked up this battery from here, a torch from here, and I am focusing it on the eye of the rabbit. And what we are saying is constriction of pupil. Okay. So when a rabbit is exposed to this kind of light stimulus, it will be showing decrease in pupil size. Another is corneal reflex, touch reflex. So here we can see this cotton twig is brought near the eye, and if you are slightly touching the cornea, the rabbit will be showing closure response. Okay? So we are actually performing this kind of experiments. We are trying to study the effect of drugs. Another thing is very nicely this also has been articulated here is if you want to check what is the pupil size and how a drug is affecting pupil size, even that has been very nicely simulated using this particular scale. You can actually find out what is the size of pupil in terms of division. So when I'm putting the scale here, I'm finding that it is around 7 divisions is the diameter of this pupil. So like this, we can check after adding the drug how the pupil size will change. And this device called as digital tonometer is used to check intraocular pressure. Okay. So when we bring it in front of the rabbit's eye, this kind of device, which is non-touch kind of tonometer, it just creates a wave of air, and whatever is the back pressure, it is sensed by this device tonometer, and just by holding this tonometer in front of the rabbit's eye, we can check what is the intraocular pressure. Now once we understand how to check these responses, check these reflexes, we can start adding various drugs. So first step in experimentation will be to wash the rabbit's eye using normal saline. So see how all those steps have been included in all these experiments. We cannot directly start performing the experiment. We have to just wash the eye, this cornea of the eye using normal saline, and then start adding the drugs. So here facility has been provided to select the drugs, see if we are having atropine. Then you can just drag this dropper and put atropine solution in the rabbit's eye. Now this eye has been treated with atropine, and the counterpart is the control eye. Very easily we can visualize. Now we can just zoom this out and we can check how difference, what kind of difference is there in the pupil size. We can actually ask the student to check this pupil size and note the observation in their notebooks. We can also check how atropine has affected intraocular pressure. So here we are finding that atropine is not having any significant effect on the intraocular pressure. How it is affecting response to torch. See, the control eye is still showing this reflex to light sensation, but the atropine treated eye is not showing any response, which is cycloplegia. Now you can imagine the ease of teaching pharmacology using such kind of simulations. In wet labs, it is very difficult to even demonstrate cycloplegia kind of thing where the rabbit's eye is not responding to a light reflex. Okay? But using this particular simulation, we can very efficiently teach our students what is really happening. And then teachers will be having time to explain to their students what is the actual mechanism, why actually atropine is causing paralysis of iris muscles, on which muscles it is acting, which receptors are involved. So if we are having this simulation, the practical hours will be very pragmatically, very nicely used for telling the students regarding the theoretical aspects of this practical. Okay? So this is how all the responses of different drugs have been simulated here: atropine, ephedrine, physostigmine, lignocaine. So if we use lignocaine, we will be able to show the students how local anesthetics are inhibiting the touch reflex, corneal reflex. Okay. So whatever experimentation you want to design, you can do it in this. Suppose I want to use the same rat, then what I will be doing, I will be just washing the eye with saline, and whatever residual drug is there, it will be washed off, and then I can add the next drug into the same eye or the another eye. Like this, whatever a teacher wants to demonstrate, he has been enabled, he has been empowered to tell his students how actually the experiment is to be demonstrated, and how he can very well explain to his students what all kind of responses we are recording here. See, when we learned pharmacology or this experiment, rabbit experiment, which we were just having books and journals and 1 or 2 demonstrations per year. And demonstrations like tonometers were totally impossible for us. But using this particular simulation, we can make our practical hours wholesome. We can tell the students what all is being done in this experiment throughout the world and how it is helping us to understand the pharmacology. Let me show you one more experiment. This also has been devised by Dr. R. Ravindran sir, and Dr. R. Ravindran sir has won many prizes for this design of this particular software. Effect of drugs on blood pressure and heart rate. This is considered as a very nicely designed software and has been accepted in all the countries throughout the world. Means people, pharmacologists, teachers are using this particular software package, particularly this experiment, to show the intricacies, the complexities of effects of drugs on this dog blood pressure and dog heart rate. Because there are many experiments in autonomic nervous system related pharmacology where, say, we want to demonstrate nicotinic effect of acetylcholine on blood pressure. If you want to teach this using conventional methods, it will take hours together to explain to students how it is done. Fortunately, we were having actual wet labs on dog blood pressure, but there also it was very difficult to understand all these theoretical aspects related to the experiment. So nicotinic effect of acetylcholine, I came to understand or really understood it when I used this simulation for the first time. Let me show you a simple demonstration of Dell's vasomotor reversal. Okay, so we know that epinephrine is having, let me inject epinephrine here. So epinephrine gives biphasic response. So pharmacology teachers will appreciate how difficult it is to convince our students what is meant by biphasic response. Okay? Here easily, if you are having the simulation with you, you can easily tell that initially there is rise in blood pressure and then there is slight fall in blood pressure. Now again, here we will be having time to tell our students that alpha effect is a short duration effect, beta effect is comparatively prolonged effect. That's why we are having initially rise in blood pressure and then fall in blood pressure, because epinephrine will be affecting both these types of receptors. Okay. Now we have to tell our students what is meant by Dell's vasomotor reversal. So I am administering an alpha antagonist here, pentolamine here. So what pentolamine will do, as it is causing relaxation of this blood vessel muscles, smooth muscles, it is causing fall in blood pressure. And again, the blood pressure is coming to normal when this entire drug is distributed in the vasculature of animals. Now, what we are having here is a dog which has been treated with a large dose of alpha antagonist. See what dose was given? It was 1000 microgram per kg. At this dose, all the alpha receptors in animal's body will be blocked by this alpha receptor antagonist. Again, if suppose I repeat the same dose of this epinephrine, this adrenaline, this adrenaline dose is repeated again, same dose of 2 microgram per kg I am repeating. And then let us see what happens here. What we are seeing is a very distinct response, very different response from what we had recorded earlier. Okay. Earlier, we had seen biphasic response of adrenaline, but when the dog was treated with large dose of alpha antagonist, what we could see is only fall in blood pressure that was comparatively sharper than only adrenaline related effect. Now here we can have time to tell our students how for the first time this experiment was performed by Sir Henry Dell, which animal he was using, how he came to know that ergot alkaloids are really affecting certain receptors in the body, which he named as alpha receptors. This kind of historical aspects we can tell our students, and then students will be able to really grasp the meaning of Dell's vasomotor reversal, how the discovery of alpha beta receptors came into existence, and how easily we can demonstrate this particular types of responses. Similarly, we can demonstrate nicotinic response of acetylcholine, which is also a very complex kind of experiment. But using this conventional classical pharmacological setups, our earlier years pharmacologists, veteran pharmacologists, they have discovered many things related to pharmacology. So if such package is available with us during practical hours or even during theory hours, we will be able to very nicely demonstrate to our students the real meaning of pharmacological experiments. Okay. So in this dog blood pressure experiment also, around 12 drugs have been demonstrated. We can use permutation combinations of them again and enable our students to understand complete autonomic nervous system related aspects. Okay. Then there are certain aspects like physiological antagonism, right? Where if you administer adrenaline in the beginning and then if you administer histamine, the effect of histamine will be masked to some extent. This is what we call as physiological antagonism. So such concepts also we can very efficiently teach to our students if we are using this particular package. Now, as this comes from a very experienced pharmacologist who also is well versed with designing of the softwares. So this is a rare combination. This rare combination happened in case of Dr. R. Ravindran, who is having interest in computers, programming, as well as he is having an in-depth knowledge of pharmacology. So when such a person develops some simulations for his students, and this has not been done within a day, he has taken years of efforts. Many pharmacologists were involved in validation of this particular software, and then this particular package has come up. So such kind of software packages, simulations to teach pharmacology are possible when pharmacologists, teachers spent decades together for development of some simulation. So we can understand the value of such simulation in teaching and learning pharmacology. Okay. Apart from this, in this particular package, we have added many demonstrations, video demonstrations, high quality video demonstrations for experiments. Say frog heart is one experiment, then guinea pig ileum is another, rat ileum, rat stomach strip, rat anacoccius muscle, then rat tracheal chain, then rat uterus. All these demonstrations we have included in this particular software package. I'm just showing you one small demonstration which is related to isolation of frog heart from the frog's body. Okay, so this kind of demonstrations are very rare again. This high quality, high definition videos have been created where we are now demonstrating how to remove pericardium, okay, from the frog's heart. And then the entire process of cannulation of sinus venosus has been demonstrated to the users in as a high definition video. So you can see the quality of this and the clarity with which the demonstration is happening here. Okay? So once students start using such high quality material, they will be sure developing understanding, better understanding of the subject, and they will be developing liking also regarding the experimental pharmacology. So once we do such, or once the student learns how actually the heart is being cannulated and how actually it is being taken out of the frog's body and mounted on the stand, then the user will be having the real feeling of the simulation. Okay? The user will be able to understand what kind of instrumentation was used, how the setup appeared, how actually the heart was isolated from the frog's body and still it continued to work outside of the frog's body. So once this kind of demonstration is given to student, he will be then taken to actual simulation where see all those minor things like what is meant by a systole, is how a systole will appear in this particular experiment, how a diastole will appear. Then when we are adding different drugs, how that drug solution or where actually the drug solution is added and how that drug solution is affecting the cardiac activity. These things have been very nicely created, nicely simulated using this particular. This is what like was being performed in wet laboratories. Okay? Unfortunately, our students are deprived of this kind of experiments now, UG students at least. But using this software, we can compensate for the loss of wet lab experiments which are happening. And using this simulation, teachers are having a very powerful tool to properly explain experimental pharmacology to their students. Now, each time when we take effect of a drug, we have to record normal responses. So, actual wet lab related precautions are also being taught to students. They are being reinforced through such kind of software package. Okay? So you can easily understand if such a tool is available, how nicely we can teach pharmacology to our students. Okay? And then when we are using in this particular experiment, if suppose we are administering a very high dose of calcium chloride, in which particular phase of its beating the heart will stop. So all these things have been demonstrated or simulated in this particular software. High quality videos and very high quality simulations where students are actually interacting with the setup which is there on the screen. Okay. Apart from this frog heart, we have also included multiple bioassays. In this software package, there is bioassay by matching method, bioassay by bracketing method, then interpolation type of bioassay, multiple point bioassay, all these types of bioassay have been added. And then teachers can again keep on changing this doses which are being used and the way experiment is being performed, or the teachers can give different challenges to the students to use a different dose range, different kind of you can say doses, different kind of experimental designs, and really try to understand how a bioassay is performed. Okay? So the beauty of this particular software package is when you record the responses here, you will be able to count the height of responses as it is done in the wet lab. Okay? So I'll show you how it is done actually. When we go to observations, see here, whatever responses we have taken, they will be recorded. And using this scale, now you can count the height of this response and you can enter it in the observation table. Okay. So there are many complex concepts involved in this bioassay kind of experiments or DRC related experiments, which are difficult for students to understand if there is no proper simulation available. Okay. So we can go on selecting different doses or different setups like this, and again we can check how actually the selected dose is working in this particular setup. If suppose we give another double dose of it, how it will appear, that also we can select and show here. So let me select now double the dose of selected standard dose. Again give a wash, and we will see how nicely, how easily we can demonstrate bioassay. This is matching type of bioassay here. Okay. And then students can learn the principles of bioassays, as well as they will be having thorough understanding about how calculations are being done, because calculations many times they have become very challenging for the students. So here students will be able to use these various dose combinations, and they will be taken to the way in which actual concentration of the unknown is calculated. Okay. So once this double dose of standard and double dose of test have been done, we can go ahead to observations. We can put these observations and calculations will appear here. Okay. So like this, we are really able to efficiently tell our students how bioassays are performed. Okay. This is something unique. This we can call this as an empowerment of pharmacology teachers, because in earlier days, a decade before, when we tried to teach these experiments in absence of experimental animals or actual wet lab experimentation, it was becoming very difficult for us. Okay? So this is how we can show our students the calculation and what was the ultimate concentration of unknown drug. Again, in this particular software package, we are given MCQs. So apart from seeing the demonstration, apart from performing the experiment, students can answer the questions on screen and they can check what is their performance. Okay. Then another nice thing about this is whatever student has done on screen, a detailed report of it will be created. So if teacher wants to see what his student has done, how the student has performed his experiment, then very nicely all those things which have been done by the student will appear on screen here. And this particular printout we can put as journal for the student. Okay. We can ask the student to just take out the print of whatever he has done in the screen and put it as a record of laboratory work the student has done. So creation of these reports, and again here you can add the name of the student, your institute's name, and when that particular practical was performed. Everything will be recorded directly. And then in the teacher's account, this particular report will be saved against the name of the student. So you can understand how nicely the technology has been implemented in making simulated pharmacology lab 100% accurate, 100% usable for the teachers and the students. So here I demonstrated to you a simple bioassay technique of matching assay. Similarly, you can design interpolation, multiple point, all those assays are possible. Let me take you to some whole animal related experiments where we are checking or testing the behavior of animals. This is one example of electroconvulsive related anti-epileptic activity. If suppose you want to demonstrate this experiment to a batch of 25 students, many times students will not get a chance to understand what is meant by clonic convergence. Okay. But here, using this particular software package, you can tell your students, see, when the mouse is showing this kind of response, you have to call it as a clonic conversion. Okay. And when the mouse is going in this kind of conversion, whereas it's extending its hind legs and taking its forelimbs near its chest, that should be called as tonic conversion. Okay. So this tonic clonic conversion difference you can just demonstrate efficiently to your students using a couple of clicks on your mouse. Okay. In this experiment now, you can administer the drug to animals, which is demonstrated here, and after administration of a drug, after 30 minutes now, even that time lapse will be simulated here. Means when we inject the drug, a time lapse will be shown. After 30 minutes, we will be actually testing the animals on electroconvulsive meter. Okay. So let's select one animal from here, apply electrode to it, and then give a shock to this animal, and you will see all phases of responses, and then you will be getting a chance to record different phases and their durations. So I'm giving shock to this animal now. As soon as animal goes into tonic phase, I'll be checking for how long the animal will be in the tonic phase. Okay. So according to me now, almost for 14 seconds the animal was in tonic phase, and then from 14 to around 26 seconds the animal was in clonic phase. It is in stupor, means it is not showing any other response, it is not even moving on its own. But when it is teased by the experimenter, it will be then moving to some extent. So that is the duration of stupor. Okay. It's possible that if the shock is not tolerated by the animal may die also. So all those kind of reportings have been added, whether the animal recovered or not. Okay. See, when we are removing these things and when we are just teasing the animal is becoming active again. Okay. So animal has not died. Like this, all our observations we can record, and then how the drug treated animals are behaving, that we can check. So whatever we have done here, whatever observations are there, all those observations will be automatically carried to the observation table. And this is how students will come to know how to actually perform this experiment. Beyond this, whatever difficult terminology is there, like what is minor tranquilizer, analytics. So we have included glossary related to each experiment here. And then scientists who have worked in the area of epilepsy and what are their discoveries. Even these things have been added to this software package. Okay. So now you can understand, means starting from history to theoretical details in pharmacology to how to perform practicals, very high definition demonstrations of those experiments, simulations for actual interactions, and real engagement of the learner into this particular experiment. All these have been taken care of while preparing this particular software package. Apart from this, we have also added exam mode in this particular software package. So what actually you have to do is, when you go to MyCalPharm, and if you log in into this MyCalPharm, you will be having practice exam here. This practice exam is for students now. Students can perform the practicals multiple times.
Patil sir,
Yeah.
just a minute, sir.
Yeah.
Sir, actually, good you are talking about the exam mode. The participant should know about it, but I need to share it with you that the current arrangement with the institution is only for the tutorial mode. But I would means it is good means we can talk about it how we can add exam mode to the institutions. But I think that would be great if you can yes talk about it. But for your understanding and for participants' understanding also, currently, sir, the arrangement is only for the tutorial mode. But
Okay.
let us have a look what value it gives, and then maybe how we can add it. We can talk about it later.
All
So,
right.
sir, sorry for interrupting, sir.
No, no
Please, sir.
problem. So, see, this exam mode is also just, I'll be taking 1 minute to demonstrate what exam mode really gives us. So, here the task which has been given to student in the exam mode is to check effect of drug on rabbit eye and identify which drug it is. So, when you perform experiment, the student will be performing this experiment. He will be taken to same kind of setup, but he will be provided with an unknown drug also. Okay. And the same will be done in case of bioassays, where the student will be provided with an unknown solution, test solution, which is not the concentration of which is not known. So the student will be performing bioassay and he will be submitting for evaluation purpose. So even this particular aspect of evaluation of the students has been added. Now it is possible that if you really want to evaluate how your students have learned, you may prefer to go for this particular exam mode also. But yes, this is also a very nice thing which has been prepared. Now, another beauty is when two different students are performing this experiment, they will be getting different unknown drugs. It is not that only single unknown drug has been simulated here or the single concentration has been simulated. Each student, when he performs, he will be getting a random allocation of unknown drug, a random allocation of concentration of the unknown. So here you will be having complete picture of how your student has understood the experiment and to what extent he is correctly performing it. So everything you can evaluate by seeing the report. Okay. Let me show you this one more experiment. Now this experiment we added just a couple of months ago, where our purpose was to demonstrate the experiment of anti-allergic activity by mast cell stabilization. Okay. So here also we have given a very nice demonstration about how those macrophages can be isolated from a mouse, how we are preparing this suspension, is how we are injecting ice cold phosphate buffer saline into the mouse abdominal cavity, tease it a little, collect the cells, and collect the cells by puncturing this abdominal wall, and then make a suspension of it. All these things have been very nicely demonstrated here now. Okay. So collection of this macrophages is being shown here. Once we collect the macrophages, then we go for actual simulation where the slides have been prepared. We can use this control slide and we can go for observation. So in observation now, whatever slide has been prepared, the slide is mounted on the microscope. Then we can use this focus for making the slide more clear or to see the features of the cells clearly. And then you can move this particular slide upward, downward, rightwards, leftwards, and count 200 cells. Try to see which of them are degranulated and which of them are intact. And that will be telling us what is the status of granulation. How many cells are getting degranulated by addition of a histaminergic substance. So many such nice experiments have been added. Another thing is, as we have included exam mode as well as routine experimental mode, always there is possibility that you can check whether the student is understanding the experiment properly or not. Okay? So here, the experiment of elevator plus maze has been simulated, and you have to write down or you have to just count. See, now I have started this central compartment because my animal is in central compartment. I will be going here into closed compartment because the animal has come in closed compartment. And then at the end, I will be writing the total duration of these observations. So this kind of ease and this kind of clarity is not available in any other software package which are available throughout world. Okay, this I'm saying with some authority because I have been dealing with or I have attended multiple conferences where few people demonstrated their single software package for one experiment, and the costs were in thousands. In US dollar, it was $400 per experiment. Okay, but whatever we have developed here in India using our own setups here, so we can say that this is quite relevant to our Indian population as well as international population because all these experiments we are providing at a very affordable cost, and all these details are there. One more thing I would like to share with the audience because my time is ending now. Within a couple of minutes, I will have to wind up. See, our customers are suggesting us to add other experiments in the package. We will be welcoming such suggestions. Or if suppose someone wants to add a new drug to dog blood pressure experiment, or if you want to add a new drug to frog heart rate related experiment, we're always open to that. And whatever developments we will be doing in the software, they will be provided to our subscribers free of cost. This is what we have been doing now. Almost 7-8 experiments we have added to all our subscribers as per their suggestions. Very in near future now, maybe our syllabus will be revised. Okay, and whatever experiments will be included in that syllabus. When we know about the content of the syllabus, whatever experiments are there, will assure you that if you are our subscriber, you will not be deprived of any of the experiments which we are going to add to this package. And again, if suppose you have some suggestions, if you feel that a particular thing needs to be improved, we are open to take up those suggestions also. Okay, so I hope I have given the glimpses of the richness, the content, and the way in which this particular simulation has been prepared. So I am handing over the dais to now Vikas sir for further proceedings. Please, Vikas sir, I am stopping the sharing now.
Okay, sir. So thank you, Patil sir, for taking time out and having this session at your own. So I really appreciate your time, your effort, and it is always a pleasure. Though I have heard you many times, but the way you present the whole thing, it's always a pleasure. So I, on behalf of all the participants, thank you a lot. So any questions anybody has? So one of the questions I could see onto the screen is talking about the exam mode. So all respected attendees, currently the arrangement with your institution through this group purchase, consortium purchase is only for the tutorial mode. If you think you want to add exam mode to it, you can reach out to us. We can work out something. Let us see how we can make it happen. So it would entirely depend on how many institutions are interested, and then how we can make it happen. So, but yes, exam mode is available, but the initial discussion was on these lines that only tutorial mode needs to be given. So we have done it accordingly. So one query is there, sir, Patil sir, effect of drug, we are select new animal.
Yeah, sir. Particularly, he must be talking about rabbit eye or
Yes.
dog blood pressure experiment. There, we have provided. This is really a nice question again, because what happens is, even in wet lab experiment, if suppose you administer a very high dose to a dog, the dog will be dying on the table. Okay. In such situations, what we are supposed to do, and even this has been simulated in Ravindran sir software, that if suppose you by mistake administer a very high dose, the blood pressure will go down and the animal will die there. In such case, in such scenario, you have been provided facility to add another drug, add another animal there. So you can select next animal and then start your experiment again. And then it is not that again, Sir Srinivas sir has mentioned that all students are getting same effect, which is not true. Actually, we have randomized this particular effects. So each time when you enter in the software again, when you use username and software, username and password, again you enter into the experiment, you will be getting a different effect. Now, what happens in case of DRCs is the highest response will be changing by few millimeters. As such, when you go on increasing the doses, there will be rise in response, and the response will be changing or varying by just few millimeters. So that kind of randomized effects we have added in the software already. So I request you once again to completely come out of the software, exit from the software, again enter. Next time we will be having a different set of details there. So that particular randomization has been added in here. So.
Yeah, thank you, sir. Okay. So one thing is the exam mode. As I have said, we will be discussing it. I'll be sharing the account manager's name who will be handling your accounts once we end up this session. That would also be shared into the group. And uh, so, okay, somebody's talking about X farm, so that's okay. So Srinivas sir is talking about that facing same problem in X farm. But sir, this is really different and better. And to validate my point, I would like to share with you that well, let's talk about, if we talk about the premier institutions of medical sciences, out of 21 aims, 11 aims have already subscribed to our this software, including the very reputed, the national icon aims Delhi. So we really are working hard on this, and we really, and we have now a foreign exposure also, and we have been able to take it to the
Yeah, we can.
foreign countries.
Sorry
Yeah,
to interrupt you, but if suppose Srinivas sir is expecting some more things in this, it will not be difficult for us to do. Okay, if he gives us some suggestions, we're open to take up those suggestions, and we will be working till he gets satisfied regarding the ways in which responses are coming. So,
definitely.
sir, you please let us know what kind of developments you are expecting in this. We will be doing it immediately. We'll take it as priority.
So another question is from Nimra College of Pharmacy. Is it possible to updates and advancements are continuing with the same software?
Yes, that's what we said. No? Means if suppose there are new experiment added in the syllabus, we will be adding them free of cost to all our earlier subscribers. That's our promise. That will happen.
So there would always be one version, which would be the updated version. You will always be getting the latest version. We do not have 1.0 and 2.0 and 3.0. So we will be having only one version. So whatsoever new will be added will be available to the existing subscribers complimentary. We will not be charging you any extra money for that. That is what our promise is.
True, sir.
So I think good, Patil sir. Thanks once again. Thanks for on behalf of all the participants. And now we can quickly move to the last demonstration onboarding, which is a check for plaque. So not all, but a few of the institutions have opted for this. And now I would like to Suren from our team to take us to the quick demonstration of Check for Plaque. It is India's first anti-plagiarism software, and it is being used by hundreds of reputed universities, colleges, government bodies, and we do have a unique facility. We have really developed a facility wherein we can check the documents written in regional languages. So we do have the capacity of checking any document for similarity check written in Hindi, Sanskrit, Telugu, Tamil, Malayalam, Marathi. So this is how we are. So I will not be taking much of time in introducing it and would like to invite Suren for this demonstration. Over to you, Suren.
Thank you so much, sir, for giving me the opportunity today for the demo Check for Plaque anti-plagiarism software. I will share my screen. Is my screen visible to all?
Yes, sir, and it is visible.
Thank you, sir, for confirming. Okay, sir. Everyone, this is our URL link, checkforplaque.com.in. Can you see the sign in button? You have to click at the sign in button.
Yes, madam, it is visible. You can go ahead.
Okay. So as this is for the demonstration, so I'll be using my account. So here will be the email address and the password which we have given to you all. You just have to click right on and then go to the sign in button. You see the login is success. So from here you can see the upload file. Okay, this will be the researcher name, the name of the researcher, like example, you are going to upload Vinita's report. So you can just write the name of Vinita and the email address and the languages. Example, Hindi, English, any languages you can choose from here and choose the file. Like example, this. Click on upload file. You can see the file is being uploaded and you can upload multiple files. Example, second will be another researcher name. Example, Rita. So you change the name and go for the and upload the file. Okay. And after that, you have to press confirm upload. After confirm upload, the file will be uploaded at our server. Like example, there will be here the name, the serial number, the email address of the researcher, the date and time, the submissions, the name of the researcher, report, the percentage, and you can see here, over here, we have highlight, make color of each and every percentage by different, like for the A percentage, it will be 0 to 10, for B, 10 to 40, for C, it's the orange, 14 to 16, and D will be 16 to 80, and so on, E, 18 to 100, black color. Okay, from here you can see, view the report. Over here you can see, this is CFP's logo, and here will be the logo of the institute. Like this is for Check for Plaque, so we have the logo of CFP. And you can see it will be the name of the institute, and the grade. It's a B grade, the percentage, the upload by, email address, document name, document size, time and date submissions, document language, total characters, and this is the barcode where you can scan and check the file whether it's the same or not. Like sometimes some of the researchers who just they will change the percentage of the file. So we have given a barcode so you can't change the file's percentage. And this will be the file. You can see. Okay, this is the multiple color you can see here. This is the colors of the file which you have taken from the website. Example, we can see this is brown color. 5.5% is being taken from this URL number. And you can see we also have an image. Right now, this report doesn't have any image, so it's for you a 0. And you can see there's a percentage of the similarity is showing below than 20%. Is it okay for all? So the second, we can go to analysis. In here, we can manage the file size. Like example, when we click over here, you can see the update percentage and the original percentage is the same. But some, college or university or institute, they need the report below than 1%. So what you can do here, you can check from here the URL that similarly less than 1%. Example, this is 5%, this is 2%, and this is 3.57. So you can just click over here and you can view change. So you see now the 5% is 7.782. And you can just apply the size to PDF. And you can see now the percentage of the file is showing 7.782. So from here you can see now it's changed. See, only showing you two URL color, that is brown and pink, because this is above than 1%. So over here you can have an institute details where you can check that how many submission you have been used. Like example, I have 3 uploads and right now I haven't used any upload, so the total is still here, and my submission date, my end date, my total upload, and you can click at that and you can see the users list in case you have any users. So right now I am only the users, so it's only mentioned my name over here and my email address, my mobile number, and register date, whether I'm active or inactive status. So you can see that over here I have used one user. Total uploads is only 1 and active users is only 1. You can add from here. If you want to add the user, you can put your institute name and how many total uploads you would like to give, and the name, the mobile number, and the gender, and you can create the password, and you will save, and you will get the email by the name of the person on the mail ID, the user ID password. Anything else on this? You can see we even have a logo over here by that institute name, and it also show a logo of our institute. Is it all okay? Hello?
Yes, sir, you're audible.
Okay. Sir, that's all for today. Any question, answer?
So this was a demonstration of check for plaque by my colleague, Ms. Soren. And though not many colleges have opted for, but whosoever has opted, we have already activated the account. We have shared the login credentials. And in case you need any queries, if you want to ask anything, please feel free to ask any questions pertaining to this. One of the questions which which need to be answered is about the IP access, IP address for accessing. So let me give you some in-depth clarity about what IP address access is. So IP address is, you can say, is some numerics provided by your service provider, be it BSNL or be it Airtel or be it Reliance Jio or whatsoever service you are using at your location. Now, just to access resources from the outside world, particularly the cloud-based services, they provide you a static IP and your IT team makes an arrangement that whensoever you hit on to any other server for accessing content, then that public IP should represent your institution. Now, how it works in a very layman kind of language, once you share that static IP with us, we whitelist it in our server. And the same IP can be used for accessing multiple products. Like you have, somebody has asked into the chat that is the same. Yes, whether you are using it for accessing journals, whether you are accessing it for any database, any software, legacy drug, whatsoever. The static IP, if the only thing you need to be careful about is it shouldn't be the dynamic one. If it is dynamic, then the challenge would be there. It will keep changing every time. The server will receive a new IP, and once it will not match with the whitelisted in our database, the access would be denied, and you will be denied the access, which would create a problem. So this is about the accessing through IP for all the product. Whosoever institution has subscribed for what, we have given the login credentials already. In fact, for this check for plague and MyCalPharm. If you want it to be also on IP, we can make it, but for the ease of access, we are given it so that you can access it at any point of time. So any questions pertaining to, like, because the demonstrations, the onboarding is complete. We have shown you the products, how to make use of it. And as I have promised in my earlier, in the morning when we started this session, that we will be conducting more such programs for optimization of the resources subscribed. You should get the value for what the institution has decided to subscribe for you. So any questions, you can unmute yourself. I'll be happy to answer those to the best of my knowledge and information. And if there is anything you want to write it in the chat box, I'll be happy to read it from there also. So,
hello, sir.
Yes,
sir,
yes,
this
please.
is Dr. Shivashankar Reddy.
Yes,
calling from Shanti Pharmacy, sir.
Right.
Sir, when we do this plaque check,
right.
So maybe within one time, the things doesn't go well, like within the limit.
Okay.
When we upload for the second time.
Yes.
So
your question is once you update it
server?
and you upload it again for checking. So, sir, that would be counted as a fresh submission, if that is what your query is.
Yes, sir. And apart from that, one more thing, small clarification, sir.
Yes, sir.
When we upload for the first time for the plaque check.
Right, sir.
So, second time when we upload, the content is not put into the web so that it is again taking it as the already submitted one, and we may get a higher percentage of plagiarism. So is there any option where we need to select the button that it is not put into the internet
database
for the first time when we upload?
Database.
So
Correct.
please give.
Correct.
Yes,
So I
please,
got your query, sir. So yes, as a standard, when you do a check for the first time or a second time, whensoever, sir, we do not put it in the database directly. Or for that reason, we do not check it from the database unless otherwise you specifically instruct the software to do so. So Dr. Sivasankar, what I want to say with this, and we have a provision, we have a checkbox. Once you will instruct, you will click on that checkbox that yes, we want to check it from the earlier uploaded documents, earlier uploaded dissertations, thesis, then only it will check, sir. Otherwise, by default, it won't check from the earlier uploaded documents, sir. Does that make sense, sir? That resolves your query?
Yeah, yes. Thank you, sir. Thank you.
Yes, thank you, sir. Thank you, sir. Now, let me quickly. Yeah, sorry, somebody saying. Please, sir.
Good afternoon, sir.
Yes, sir.
So myself, Naresh Babu. Sir, in the plagiarism check that AI is disabled, the option.
Yes, sir. Actually, we
and
do
it is
have
very important that nowadays we need to know that AI percentage also.
I understand, sir, and I really respect that, and I value what you are coming from. That is what another challenge for everybody is. Like nobody wants to do anything now. They have a assistant with them using the AI and getting the work done. But again, really the arrangement which has been done with the colleges for this particular software is that AI is not as of now part of it. If we need to, if you think you need to add that, then again we have to work it out. We can look into the possibility how we can do it. But current arrangement is without AI. We have the capacity. Like if you have attended, so we have, like in the case of my farm, so we do have exam mode, but the arrangement is like that only tutorial mode is being. So we have that capacity of checking it for the content generated through AI, but that arrangement has not been opted by the colleges when we were like negotiating, discussing it with the team. So, but anyways, I take your point, Dr. Naresh, and we will see what can be done. So for that, I request you can reach out to us, let us work out something. Right, sir?
Okay, sir. Thank you.
Okay, sir. So I'm assuming that there are no more queries. I would request Ruchi. Ruchi, can you please share the that contact us sheet so that if somebody wants to take a screenshot or click a picture, they can take. So we are providing you 3 different projects for which we have demonstrated. So I want to share one document, not document, means a contact us page wherein you can reach out for each of the project. Anything, though everybody can help you, including me. But we have some arrangement over here for up-to-date Lexi drug. Your point of contact can be Miss Ruchi. Her name, mobile number, and email address are there onto your screens. Same way, you have the Michael Farm. It is for a couple of people are there. Miss Hema is there. Mr. Shankar is there. Their phone numbers and email IDs again there. And finally, check for Plag, which is the plagiarism software. So Miss Sorenkor is there. Her number and the email ID is there onto your screens. You can make a photo, click it, and I'll be also posting it into the group. And yes, we all means we are into a common group. You face any challenge, we are available on phone, email, WhatsApp, howsoever it is convenient for you. We'll be happy to serve you, and we promise you to provide best of our services. One query is there. Let me answer that. The query is there any limit for check for plagiarism documents for a day? So the current package which has been opted by your institution is for 400 submissions. So, and the size of each submission is 10,000 words. So for that reason, you can check up to 400 submissions, and it is an annual basis. So no, we don't have any limit for a day. But yes, your account is limited to 400 submissions in a year for the complete year. Now it is up to you. You want to use it in a day, in a month, in a week, or in a whole year. So this is how we have designed the subscription for your institution. In case you think that the number of 400 submissions is not enough, you need more submissions, as I have said, we can always discuss. We are not adamant. We are always open to discuss the possibilities how we can serve you, so we can definitely work it out. Another question is from Dr. Sandeep. Will the plagiarism percentage of black check be similar to Turnitin plagiarism report? So all softwares are different, sir. They have different database in the background, and they have different mechanism of checking plagiarism. So it entirely depends on the sources. The big difference between Turnitin, which is a global leader, I have to accept it, and any other service provider is that they have the facility of checking from the paid content of the commercial publishers, whereas no other service provider has that kind of arrangement. And that is the reason they are quite expensive and are being used by premier institutions. But my humble submission onto this point is it is better to have something than nothing. So that difference is going to be there again, as it depends on, like if you take a complete article of Elsevier, then the difference would be high. But if the content is more from a open kind of sources, open access journals, then it would be same kind of a percent here and there. So that is, and I'm not saying it just for the sake of saying. It is based on the experience I have gained. We launched this product in India in 2017. From last 8 years, I have been instrumental in promoting it, and the kind of experiences I have had with the institutions. So I hope that answers your query, Dr. Sandeep. So now the contact us page I have shared with you.
Sir, because sir.
Yes, yes.
Sir, if possible, I would like to request Rishi to take the screenshot of even the participants list also, if possible, and provide us with that. And I don't know about other colleges, sir, but this is a recorded version. I request you to share this regarding with us.
Definitely. Definitely, we will share it with you. I humbly request for the participants. Actually, I missed that, ma'am. We should have taken a pic of the screen initially. Many people have left by now. But anyways, we still have 50 plus participants with us. I humbly request to all the participants if they can open their cameras so that we can make a record of this. We would be happy to see you all. So if that is, please, I request everyone. Ruchi, maybe you can remove the screen now. I think we have to make some changes in the settings. Can somebody help us from the team? Ruchi, can we take the screenshot?
Give me 1 minute, sir. Just 1 minute.
Please, please, please. Ma'am, Kiranmay ma'am, otherwise recording as well as who all have participated, I hope we will be having their records with us. I'll be sharing that.
Try to take the screenshots, sir, of participants. If not possible, okay, the recording, let it be there, sir.
Sure, ma'am.
Anyway,
Sure.
we'll have a record.
We can.
I'll claim it for having a training session, for the next these things, sir.
Yeah, surely.
Will to be processed.
Surely, ma'am. Surely we will do. No problem. I have that arrangement now. I can take the pic Ruchi, I'm taking it. Don't worry. I'm on to screen 2 now. Just a moment, ma'am. I'll be doing it.
So we have taken it. Selfie.
Okay. All the 3 pages you have taken, Ruchi? There are 3 pages.
Yeah, give me.
Please check, because there are 3 pages I can see. If there are 3 pages you have taken, then it is fine. Though many people have still not opened their cameras, but that's okay. Maybe you can take, 3 pages.
Sir, we have taken it. We have taken it for 3 pages.
Okay.
It's done.
Thank you. Kiranmai ma'am,
Okay, sir. Thank you.
if you want to say something to the participants, I'll be happy.
No, nothing, sir. You have done it from all our side. Thank you very much. Thank you very much.
So once again, I thank everybody who has joined in, though many people have left. Thank you for your time. It is a start of a relationship, and I assure you that we will be giving more value to you in terms of more such solutions. Somehow, though I'll be honest, I haven't planned for it, but somehow the life has been like this, that we have been working into the pharmacy segment. I proudly say now that we are the one with the maximum number of digital solutions under one roof, and that has given me more strength. And I look forward to your submissions, look forward to your feedbacks to make this effort more worthwhile, more in line with what your expectations are. So thanks once again to everybody who has joined in. Thank you, Kiranmai ma'am, for making this happen. Padmanabha sir, I don't know whether you are there in the group or not now. So thank you, everyone. And we are concluding the session here.
Right, sir. Thank you, sir. Thank you very much.
Thank you, ma'am.
Thank you, sir. Please share the recording, sir.
Definitely, ma'am. We will.
Yeah, thank you, ma'am. Thank you, ma'am. Thank you all the faculty and
very much.
principals.
Right.